Welcome to this presentation on therapeutic enzymes, from benchside to bedside. I am Dr. Suresh.S, Assistant Professor in the Department of Biotechnology, Faculty of Science and Humanities, at SRM Institute of Science and Technology, Ramapuram, Chennai. Our learning objectives for today are to understand the basic concept of enzymes, recognize their roles in health and disease, relate genetic defects to enzyme deficiencies, learn examples of enzyme-based diagnosis and therapy, and finally, appreciate how enzyme research translates to bedside use. Beginning with our introduction, enzymes are protein biocatalysts that regulate metabolic reactions by lowering the activation energy. Their activity depends critically on proper protein conformation. Dysfunction leads to metabolic or genetic diseases. Enzymes have been applied in biomedical fields for centuries, starting with the discovery of amylase. Pepsin was identified as the first natural therapeutic enzyme, and alteplase was the first recombinant enzyme. Looking at the mechanism of enzyme action, enzymes accelerate biochemical reactions without being consumed or altering the chemical equilibrium. As shown in the diagram, the substrate enters the active site of the enzyme. The enzyme-substrate complex forms, with the enzyme changing shape slightly as the substrate binds. The substrate is converted to products. The products then leave the active site of the enzyme. Enzyme production and activity are regulated based on physiological needs. Substrate binding to the active site induces a transition state, forming the product and releasing the enzyme. Enzyme activity is controlled by feedback mechanisms and covalent modifications, such as phosphorylation. For example, the enzyme glycogen phosphorylase is activated by phosphorylation when epinephrine binds to muscle cells, catalyzing the conversion of glycogen into glucose 1-phosphate. Enzymes are classified into 6 major groups based on the reaction catalyzed. These include oxidoreductases, which catalyze oxidation-reduction reactions, such as dehydrogenase; transferases, which transfer a functional group from one substance to another, like kinase; hydrolysis, which form two products from a substrate by hydrolysis, such as lipase; lysis, which perform non-hydrolytic addition or removal of groups, such as decarboxylase; isomerases, which perform intramolecular rearrangement, like mutase; ligases, which join two molecules together with the simultaneous breakdown of ATP, such as synthetase. Beyond digestion, enzymes are indispensable for a wide range of functions, including DNA replication, energy production, muscle contraction, and detoxification. This slide highlights examples of enzymes of the human physiology. For instance, superoxide dismutase protects cells from harmful effects caused by superoxide anion and other free radical reactive oxygen species. Dioxygenase catalyzes two oxygen atoms onto a substrate without the reduction of one oxygen atom from dioxygen into a water molecule. Now, let us discuss the one-gene one-enzyme hypothesis, proposed in 1941 by George Beadle and Edward Tatton. This hypothesis states that, in general, each gene directs the formation of one enzyme. They identified bread mold mutants that were unable to make specific amino acids because a mutation had broken a necessary enzyme. Their two-step experiment involved exposing Neurospora crassa spores to X-rays to induce random mutations. The resulting mutants were grown on minimal medium supplemented with either the full set of amino acids or the full set of vitamins. By identifying which single nutrient rescued each mutant, Beadle and Tatton could link a specific gene mutation to the loss of a single enzyme required for a single step in a biochemical pathway. Inborn errors of metabolism, or IM, were first described by Sir Archibald Garrod through alkaptonuria, identifying its recessive inheritance and metabolic defect. IMs are inherited single-gene disorders caused by defects in enzymes, cofactors, or transporters. Mutations cause enzyme dysfunction or loss of production. These errors lead to either toxic substrate accumulation or a deficiency of essential metabolic products. Examples of inherited metabolic disorders include phenylketonuria under amino acid metabolism, von Gurk's disease under carbohydrate metabolism, and hemochromatosis under metal storage disorders. Phenylketonuria, or PKU, is a hereditary metabolic disorder that impairs the body's ability to metabolize the amino acid phenylalanine. The enzymatic function is normally performed by phenylalanine hydroxylase, which catalyzes the conversion of phenylalanine to tyrosine. In pathophysiology, a deficiency or malfunction of this enzyme results in phenylalanine accumulation and reduced tyrosine levels. Elevated phenylalanine concentrations can reach toxic levels, posing significant health risks. PKU follows an autosomal recessive inheritance pattern. The diagrams contrast the normal pathway, where phenylalanine is converted to tyrosine, with the abnormal pathway, where the deficient phenylalanine hydroxylase causes phenylalanine to accumulate and subsequently be converted into abnormal products like phenylacetic acid. Other significant inborn errors of metabolism include galactosemia caused by the deficiency of galactose 1-phosphate uridyl transferase, leading to galactose 1-phosphate buildup and tissue damage. Von Gerk's disease, deficiency of glucose 6-phosphatase causes glucose 6-phosphate accumulation in the liver, resulting in fasting hypoglycemia. Wilson's disease, a mutation in the ATP7B gene that impairs copper transport, often identified by Kayser-Fleischer rings around the cornea. Type 1 hyperammonemia, a urea cycle disorder due to carbamol phosphate synthetase I deficiency, causing high blood ammonia levels. Blocked metabolism due to enzyme dysfunction occurs when a deficient or defective enzyme creates a metabolic block in a pathway. This block causes one of two issues: toxic accumulation, the unprocessed substrate builds up to harmful levels. For example, in PKU, the mutation in the PAH gene causes phenylalanine buildup, leading to neurological damage. Essential deficiency, the required product is not formed or is produced in insufficient amounts. The diagram illustrates this block in the phenylketonuria pathway. A deficient phenylalanine hydroxylase prevents the conversion of the phenylalanine substrate into the tyrosine product, resulting in both toxic accumulation and essential deficiency. The metabolic block in PKU causes an essential deficiency of tyrosine, which is needed for synthesizing catecholamines like dopamine. For diagnosis, the Guthrie test is used. Its principle is to detect elevated phenylalanine using beta-2-phenylalanine, which inhibits the growth of Bacillus subtilis. A positive result occurs when high phenylalanine, above 2 mg per deciliter, neutralize this inhibition, leading to visible bacterial growth around the blood disc. Enzymes are crucial in clinical diagnostics, as seen in the list of serum enzymes in disease diagnosis. For instance, aspartate aminotransferase, or AST, is used in diagnosing myocardial infarction and liver conditions. Alanine aminotransferase, or ALT, is primarily used for diagnosing viral hepatitis. Amylase and lipase indicate acute pancreatitis. Serum enzymes act as a chemical fingerprint for damaged tissue. When an organ is damaged by infarction, infection, or trauma, the cells break open through necrosis, releasing high concentrations of their specific internal enzymes into the bloodstream. Organ-specific diagnosis allows clinicians to narrow down the problem. For example, high levels of alanine transaminase, or ALT, are highly specific for liver disease. Similarly, creatine kinase MB, or CKMB, specifically indicates myocardial infarction or heart damage. Differential diagnosis uses enzyme patterns, such as high alkaline phosphatase and gamma-glutamyl transferase together, to suggest biliary obstruction. Enzymes are also used for monitoring disease progression and therapy. For example, serial measurements of amylase and lipase track the severity and resolution of acute pancreatitis. The magnitude of the enzyme elevation also correlates with the extent of tissue damage, aiding in assessment of prognosis. Finally, the diagnosis of inherited metabolic disorders involves screening for a deficiency or absence of a functional enzyme, which is the primary cause of the disease, such as detecting the absence of galactose 1-phosphate uridyl transferase to diagnose galactosemia. Therapeutic enzymes are biocatalysts used to treat medical conditions and diseases. These treatments are able to convert a wide range of target molecules to restore the correct physiological metabolism. Therapeutic enzymes offer unique benefits. They exhibit immense catalytic power and high substrate specificity, meaning a single enzyme molecule can convert multiple molecules of its substrate. They are used to either break down harmful substances or deprive diseased cells of essential nutrients, providing therapeutic mechanisms not possible with traditional pharmaceuticals. The evolution from bench to bedside is based on the fact that enzyme metabolism is a foundational bioprocess crucial for human functions like digestion, detoxification, and supporting innate immunity. Abnormalities in this process cause several metabolic disorders. Molecular biomarkers such as alanine transaminase and lactate dehydrogenase are used for diagnosis. Recent advancements in recombinant DNA technology, protein engineering, and nanotechnology have provided an astounding platform for developing enzymatic drugs. Therapeutic enzymes have varied applications, as illustrated in the classification. In cancer treatment, they are used as anti-cancer agents, such as asparaginase and arginase. For clotting, they act as fibrinolytic agents, including urokinase, streptokinase, and nattokinase. For genetic disorders, we see use of enzymes like aldosterase, phenylylase, and pagadamase. They are also used for gastrointestinal diseases as digestive aids, such as lactase and amylase. Enzyme replacement therapy, or ERT, is often the only available treatment to replace a missing function and prevent progressive organ damage, particularly for inborn errors of metabolism and lysosomal storage diseases. ERT involves the intravenous administration of the correct enzyme, often produced via recombinant DNA technology. This enzyme is typically modified, for example, pegylated to allow for a longer half-life, more potent activity, or targeted delivery. The first successful ERT were for alpha-1 antitrypsin deficiency. The diagram shows the simple mechanism of ERT. In the disease state, a deficient enzyme leads to toxic, accumulating substrate inside the lysosome. In the therapy state, an IV infusion delivers the engineered enzyme, which restores function by metabolizing the accumulating substrate. Enzyme replacement therapy is critical for many genetic disorders. For Pompe disease, ERT with AT-GAA is shown to restore autophagy and improve muscle function. In Gaucher disease, an inherited deficiency of lysosomal acid, beta-glucocerebrosidase leads to the accumulation of substrate in the liver, spleen, and bone. Recombinant forms of glucocerebrosidase have been developed and are now the standard of care. For Fabry's disease, ERT replaces the deficient endogenous enzyme with recombinant agalsidase alpha or agalsidase beta. Other therapies include chaperone therapy, which uses a molecule like miglustat to stabilize the endogenous enzyme, and substrate reduction therapy, which inhibits the synthesis of glucosylceramide to reduce the accumulation of glycosphingolipids. Enzymes function as indispensable markers in disease diagnosis, including in oncology. Acid phosphatase, AP, or PAP, is a highly specific indicator for prostate cancer. Cathepsins are lysosomal proteases whose overexpression links to tumor invasion, proliferation, and angiogenesis in cancers like breast, colorectal, and lung cancer. Lactate dehydrogenase, LDH, reflects abnormally high metabolism glycolysis, which supports malignant tumor growth in various cancers. Thymidine kinase 1 or TK1 activity is a good indicator of cell proliferation and malignancy across various solid tumors and blood cancers. A prime example of enzyme therapy in oncology is the use of L-asparaginase. Normal cells can synthesize their own L-asparagine using asparagine synthetase, or ASNase. However, many leukemic lymphoblasts, specifically all cells, are deficient in or lack sufficient ASNase expression. Because these malignant cells cannot produce L-asparagine de novo, they rely heavily on the circulating extracellular supply. By administering L-asparaginase, we selectively deplete the circulating L-asparagine, effectively starving the malignant cells, which inhibits DNA, RNA, and protein synthesis and ultimately induces apoptosis, or programmed cell death. Enzyme-based biosensors exploit the natural specificity of enzymes for their substrates. The most common class utilized is oxidoreductases, as their reactions often involve the consumption or production of electrochemically active species. Examples include glucose oxidase, or GOX, used for glucose monitoring, such as in glucometers. Cholesterol oxidase, or CHOX, used for cholesterol detection. Lactate dehydrogenase, or LDH, used for lactate detection. Horseradish peroxidase, or HRP, often used in signal amplification for various assays. Another class is hydrolysis, which includes urease for urea detection and acetylcholine esterase, used to detect pesticides and nerve agents which act as inhibitors of the enzyme. The mechanism of biosensors involves two fundamental steps: biorecognition and signal transduction. Biorecognition is the enzymatic reaction where the immobilized enzyme interacts with the target molecule, or analyte, converting it into a product. This catalytic reaction causes a measurable change in a physicochemical parameter, such as the concentration of products like hydrogen peroxide, or a change in heat production. Signal transduction is where the transducer element detects this physicochemical change and converts it into a quantifiable electrical signal, such as current or voltage. Electrochemical transduction is the most common type, measuring changes in current caused by the consumption or production of electroactive species. ELISA, or enzyme-linked immunosorbent assay, is a heterogeneous enzyme immunoassay technique that uses the catalytic properties of enzymes to detect and quantify immunologic reactions. The assay uses an antibody or antigen immobilized onto a solid surface. A secondary antibody conjugated to an enzyme, such as HRP or alkaline phosphatase, is used for detection. The enzyme reacts with a colorless substrate to produce a colored product, and the intensity of the color is directly proportional to the amount of the target analyte in the sample. In the development and monitoring of therapeutic enzymes, ELISA is instrumental. Applications include therapeutic drug monitoring, to track the concentration of therapeutic enzymes in a patient's blood to ensure optimal, non-toxic dosage. Used in pharmacokinetic and pharmacodynamic studies. Immunogenicity testing, which uses ELISA as a standard method to detect and quantify antidrug antibodies that might reduce the drug's efficacy and safety. Biomarker quantification to monitor specific substrate levels that indicate treatment efficacy. Recombinant technology is essential for creating human-compatible therapeutic enzymes. Chinese hamster ovary, or CHO cells are often preferred among mammalian cell lines because they can perform human-like post-translational modifications, such as glycosylation, which is crucial for the protein's stability and proper function in the human body. Different host types offer distinct advantages. Bacteria, like E. coli, are used for economical, large-scale production due to their simplicity. Yeast, like Saccharomyces cerevisiae, offers the ability to perform post-translational modifications. Mammalian eukaryotic cells, like CHO cells, are crucial for performing human-like modifications to minimize the immune response. Let us look at a case study involving a switch in enzyme replacement therapy, or ERT, for Gaucher disease. Quebec prioritized taloglucerase as the first-line ERT for cost-effectiveness. A retrospective study involved 19 stable patients who were switched from imiglucerase to taloglucerase. The study found that while the disease parameters remained stable, a significant rate of new adverse effects occurred. 6 out of 19 switched patients reported new adverse effects, primarily infusion-related reactions such as redness and hives. These symptoms often persisted despite management attempts. As a result, 5 of the 6 patients who experienced new side effects were ultimately switched back to imiglucerase, which resolved their symptoms. This highlights the real-world challenge that switching stable patients, even to a cost-effective alternative, can lead to clinically significant adverse reactions. The path for therapeutic enzymes from bench side to bedside is a multi-step process. It includes discovery and engineering. The gene for the desired enzyme is identified and cloned into an expression vector. Host selection and development. The vector is inserted into a host, such as CHO cells, to develop a stable, high-yield factory. Preclinical studies. Extensive testing is done in vitro and in vivo to determine the enzyme's efficacy and preliminary safety. Clinical trials. Testing occurs in humans across Phase 1, safety and dosing, Phase 2, efficacy and side effects, and Phase 3, confirmation of efficacy. Regulatory approval. All data is submitted to agencies like the US FDA for approval to market the drug. Large-scale manufacturing. Production is transitioned to an industrial scale with rigorous quality control and purification. The approved enzyme is then prescribed to patients. Post-marketing Phase 4 surveillance continues to monitor long-term safety, adverse events, and efficacy in the general population. This diagram illustrates the challenges in therapeutic enzyme research and how nanomedicine is utilized to overcome them. On the left, we see the intrinsic attributes of enzymes, highlighting their strengths and weaknesses. The strengths of enzymes include high catalytic activity, specificity of interaction, biodegradability, biocompatibility, and diverse therapeutic applications. However, native enzymes face several challenges such as sensitivity to the biological milieu, a short blood half-life, difficulty permeating biological membranes, immunogenicity, and off-target accumulation. The field of nanomedicine offers a pathway to solve these limitations. By encapsulating the enzymes, we move to enzyme-loaded nanocarriers. These carriers provide several crucial benefits, including protection from degradation, preserved catalytic activity, controlled and targeted release, and prolonged circulation. For treating complex diseases, the research moves towards dual-loaded nanocarriers. This advanced strategy aims to provide a synergistic therapeutic effect, enable multidrug integrated therapy, and offer novel therapeutic strategies for hard-to-treat diseases. This chart maps the future perspectives of therapeutic enzyme development, flowing from drug delivery systems to pharmacological outcomes. The first column highlights advanced drug delivery systems, centered around smart nanocarriers. These carriers include specific types such as mesoporous silica nanoparticles, magnetic nanoparticles, liposomes, polymeric nanoparticles, and nanoflowers. The second column lists important microbial enzymes and their target diseases. For phenylketonuria, the enzyme listed is phenylalanine ammonia lyase. For antithrombotic applications, key enzymes are streptokinase, staphylokinase, and fructokinase. For anti-cancer therapy, we see asparaginase, arginine deaminase, and nitroreductase. For wound healing and debridement, the enzymes include trypsin, hyaluronidase, and collagenase. The third column details the potential therapeutic applications where these enzyme systems are used, including phenylketonuria treatment, blood clot lysis, cancer treatment, and wound healing. The final column outlines the desired pharmacological attributes that these advancements aim to enhance stability, permanence, and the therapeutic index of the enzyme drugs. In summary, enzymes are essential for life, catalyzing metabolic pathways like digestion and DNA repair, and their dysfunction is the direct molecular cause of many diseases. They are crucial diagnostic biomarkers, measuring their levels, such as creatine kinase for heart attack or ALT for liver damage, provides quantifiable evidence of tissue damage. Enzymes are a primary focus in therapy because they are highly effective as drugs due to their catalytic and specific nature. They form the basis of biotechnological tools, such as restriction enzymes and DNA polymerase. They are also used as direct therapeutic agents, including in enzyme replacement therapy or for targeted destruction of specific cells, like L-asparaginase for leukemia. Furthermore, enzymes are core to personalized medicine, as genetic variations in drug metabolizing enzymes dictate how quickly a patient processes a drug, allowing for personalized dosage. Thus, enzymes are effective both as biomarkers for diagnosis and as drugs for treatment. The take-home message is, from understanding a single enzyme's function to curing complex diseases, enzymes are the silent physicians within us. I would like to thank Dr. Harmi bin Mohammad Yusuf, Dean, and Professor Dr. Mahadev Rao, Professor, from Faculty of Medicine, UNISA, Malaysia, along with the organizing team and MBBA students. I also extend my sincere gratitude to our Honorable Chairman Dr. R. Shivakumar Sir, SRM Group of Institutions, Ramapuram and Trichy campuses, our Dean, BP Admin and BP Academic, FSH, and our HOD, Department of Biotechnology. Thank you very much for your attention.